there, we're going to talk about DNA replication. And first, we are going to start with some simple notes on DNA replication. So I have titled my notebook page, DNA replication notes. And the first thing that I need to know, pen, is that DNA replication occurs in the nucleus, right? That's where DNA is stored, so that's where DNA replication has to happen. So, DNA replication happens, oh gosh, sorry, spelling, before cell division, so that the new cell has a complete copy of DNA. So when the cell divides, we end up with the original cell and a new one, and we need that new cell to have a complete copy of the DNA. So DNA replication has to occur before cell division. DNA replication is going to follow a base pairing rule. Basically, adenine, right, which is A, will always pair with thymine, and guanine will always pair with cytosine. Because DNA replication occurs by adding one new strand, we say that DNA replication is semi-conservative because both strands will contain part of the original DNA. So, the steps for DNA replication, we are going to start with an enzyme called DNA helicase, and its job is going to be to unwind the double helix, and it is going to break the hydrogen bonds. So the hydrogen bonds that hold our bases together, DNA helicase comes in, it's going to unwind it and break those bonds. Then a second enzyme called DNA polymerase is going to bind free nucleotides to the newly broken hydrogen bonds to create that double helix. So there are free nucleotides hanging around in the nucleus. There we go. And what will happen is that DNA polymerase will start binding those nucleotides to that newly unwound strand of DNA. So let's take a look to see what happens with that. So I'm going to flip the page in my notebook. So I've got my DNA strand here. So here is my original parent strand. I can see All of my bases are connected together. And here, my DNA helix has been unwound by helicase. Right? Helicase comes in and it unwinds and breaks the hydrogen bonds that hold the bases together. So then we now have these bonds that need to be remade. So DNA polymerase is going to come in and it's going to say, okay, here's guanine. I need to match that with cytosine, right? And these are all free nucleotides just hanging out, right, in the nucleus. Then it sees adenine. It's going to pair it with thymine, another adenine paired with another thymine, guanine paired with a cytosine, and then another cytosine paired with a guanine. So as we can see, as DNA polymerase starts to add and bind those free nucleotides, it starts to recreate that double helix. So I'm going to use a piece of tape real quickly to tape down my newly formed double helix. So just put a little right there. And then I'll add another piece here on the other side. There we go. So what I have here is, right, my original parent strand. It was broken apart. So here's the original parent still. But then each new DNA strand, right, has a new copy. So we say that DNA replication is semi-conservative because this new DNA strand has a new copy and a copy of the original, and this DNA has an original copy of the parent and a new copy. So that is DNA replication, talking about the three major points, the two enzymes that are needed, DNA helicase to unwind and break the hydrogen bonds, DNA polymerase to add the new nucleotides to the growing DNA strand, and the fact that DNA replication is semi-conservative. Each new DNA strand has a copy of the original and a new copy of DNA. If you have any questions or concerns about DNA replication, please let me know.